Okay, so first of all, I'd like to thank the organizers for setting up this wonderful meeting and all of you for being here today. So the goal of the work I'll present today is to learn rules of regulation based on the expression of genes across many diverse conditions. Understanding the rules that govern gene regulation in a condition-specific manner will help us better understand the mechanisms underlying phenotypic variation and disease. Of course, the problem of studying gene regulation based on gene expression data is not new, and many methods have been proposed for addressing this task. However, there are, I believe, several reasons for revisiting this problem. First of all, the amount of available data has been increasing very fast in the last few years, with many consortia and public databases collecting data on the expression of genes across hundreds of different conditions. This increase in the amount of available data leads to an increase in modeling complexity due to the diversity of the conditions studied. When we look at a small set of related conditions, it might be reasonable to assume that each gene has a single function and therefore a single regulatory mechanism. However, this is not necessarily the case when we look across very diverse conditions, such as different tissues or different developmental stages. Finally, nowadays we have much more than just gene expression data. For example, we have information about the chromatin state in different cellular contexts, or about the interactions between proteins that act together in a complex. And therefore, we should try to incorporate this information into the modeling process. The main assumption that we make in this work is that genes are regulated by transcription factors, TFs, or combinations of TFs that recognize and bind to sequence motifs in the regulatory regions of genes. Therefore, our goal is to learn rules of regulation based on the activity of TFs across different conditions and on the set of sequence motifs that are present in the regulatory regions of genes. The rules that we learn have the form shown here. They combine a TF and a sequence motif, and they model the fact that when a TF is up or down regulated, then some genes that have a certain sequence motif in the regulatory region will also change their expression. To learn these rules, we pose this as a binary classification task, where the input is a set of genes that are up or down regulated in different conditions. And each instance of up or down regulation in this matrix is a different training example whose behavior is a function of the behavior of TFs in the corresponding condition and on the set of sequence motifs that are present in the regulatory regions of the corresponding gene. And from this input, we learn a decision tree that looks like this. And given such a tree, we can ask different questions and get different biological insights. For example, we can ask which regulators tend to be together in the same path of the tree, or which sequence motifs does a given regulator tend to be associated with, or which paths or rules of the tree are triggered by a specific subset of the genes and under which conditions. To learn this model, we use a machine learning technique called LogitBoost. And LogitBoost is related to logistic regression, and it belongs to the broader family of boosting techniques. Boosting methods can build very confident predictors using as building blocks much weaker predictors. And they do this in an iterative way. In our case, this means that we learn our tree one node at a time. At each iteration, we will go over all the nodes that we have added to the tree so far, and in each case, we will consider all possible decisions that we could add under each node. Then we will pick the combination of a parent node and a new decision that gives the best reduction to our loss function. We will add this new decision, and with each decision node, we associate a score, and the interpretation of that score is that it reflects the magnitude and the directionality of that rule. Finally, we reweight our examples so that we put more emphasis on examples that we have been misclassifying. And in the end, the score of its training example will be the sum of the scores of all the nodes through which it passes, and the sign of that sum will give us the binary prediction, up or down regulation for that example. So the model, as I described it so far, has very good accuracy. However, it might suffer when it comes to interpretability. And one of the reasons why this can happen is that with gene expression data, there's always lots of correlated features. So, for example, when we try to find the regulator for a set of genes, instead of picking the most biologically meaningful regulator, we might pick a gene that happens to be correlated with a true regulator. And this is a problem that all methods that try to model gene expression have to face. One way of mitigating this problem is by using existing knowledge about gene associations, for example, about the pathways that certain genes participate in. And there's two places in the construction of the tree where we can benefit from such knowledge. First of all, as we build our rules, we want to try to put in the same rule TFs and motifs that are known to be related to each other. For example, NF-kappa-B with a known NF-kappa-B motif. Additionally, as we construct the tree, we want to put in the same path of the tree TFs or regulators that are known, again, to be related to each other. For example, regulators that belong to the same pathway. And to do that, we start with a network of protein interactions or other types of associations between genes or between proteins, and we convert this network into a weight matrix, where the weight between a pair of regulators reflects how much we favor pairing the two regulators together. And we use these weights to weight the different rules before we add them to the tree. And as the iteration number increases, we make these weights more and more uniform, and this allows us to learn novel associations. So before I move to the evaluation of our method, I should say that the method I just presented is inspired by this previous work called Medusa. And Medusa also used boosted decision trees like we do, however it had a different loss function and did not incorporate any existing knowledge. As I'll show you later, this leads to poorer performance. So the results I'll present here are based on data from the ENCODE consortium. We obtained expression data on the expression of approximately 9,000 genes in 21 cell lines. We computed differential expression between all pairs of cell lines, and each one of these 210 pairwise comparisons between cell lines was a different column in our input matrix. We obtained the regulatory regions of genes based on ChromeHMM annotations, and ChromeHMM is an algorithm that can divide the genome into different functional units, such as enhancers, promoters, or insulators, based on the combinatorial patterns of chromatin marks. So we used ChromeHMM segmentations to obtain the enhancers and promoters of each gene, and then we scanned approximately 900 PWMs on these regions to get our set of sequence features. Finally, we selected genes that had Go annotations, such as transcription factor or cofactor, as our set of potential regulators. And to evaluate the methods, we used tenfold cross-validation. In the table here, the last column shows the average area under the curve in all 10 cross-validation folds. And as you can see, our method clearly outperforms Medusa even when we do not use any sequence features from enhancer elements and where we do not use any information about known gene associations. Adding sequence features from enhancer elements leads to an extra small boost in performance, as you can see in the third row of this table. Finally, adding information about known gene associations leaves the area under the curve almost unaffected. And this is not necessarily surprising. This prior knowledge can help us prevent overfitting and learn more accurate models, especially when we don't have sufficient training data. And this was exactly what we observed when we started artificially decreasing the amount of training data that we used. However, the main purpose of this prior knowledge is not necessarily to improve accuracy, but to learn models that are more biologically meaningful. And this is exactly the case. First, we computed the number of distinct rules that each model identified across all cross-validation folds. So fewer rules suggest simpler models, and therefore models that are easier to interpret. And as you can see, the model that uses prior knowledge, it requires much fewer rules than the rest of the models. Next, we obtained the most important features, the most important rules for each model, ranked them based on their contribution to the model's predictions, and then we looked at the correlation of these rankings across different folds. And again, larger correlations suggest models that are more consistent and more reproducible, and therefore probably easier to interpret. And again, you can see that the model that uses prior knowledge is much more consistent across folds than the rest of the models. Next, we looked at the interpretability of the models in a different way. First, we asked whether regulators in neighboring nodes of the tree tend to share common functional annotations. So here, the purple bars are the models that use prior knowledge, and the green bars are the models that do not use such knowledge. And on the y-axis, I'm showing the ratio of the number of neighboring regulators that share a common functional annotation compared to what we would expect by chance. And as you can see, even when we do not use any information about gene associations, regulators in neighboring nodes of the tree are much more likely to be in the same pathway or to physically interact than what we would expect by chance. We next asked a similar question, but this time looking at pairs of regulators and PWMs that are together in the same node. And again, we had similar results. So these results suggest that the models that we learn encode gene associations, both in their rules and in the paths along the tree. Therefore, we can look at these rules and these paths and discover novel associations. So what we did next was to identify pairs of regulators or pairs of a regulator and a PWM that occur in our models much more frequently than what we would expect by chance. We identified 160 such pairs of regulators and 54 pairs of a regulator and a PWM, and not surprisingly, the vast majority of them were already known to be interacting or otherwise associated with each other, for example, belong to the same pathway. However, as you can see, there was still a substantial fraction of these pairs that were not in our existing set of known gene associations. And as a short example, we found very strong pairwise interactions between the three genes shown here, and it turns out that all of them are important regulators of the epithelial to mesenchymal transition, which is the process by which epithelial cells turn into the much more invasive mesenchymal cells. So to summarize, we present a method for modeling gene regulation based on the differential activity of transcription factors and on the sequence composition of regulatory regions of genes. Our method can incorporate prior knowledge about gene associations, and as I explained, this is very important for learning models that are not only accurate but also biologically interpretable. This method is very generic, and although I just showed you a test application here, I would like to invite you to apply it to your own data and ask your own biological questions. I didn't have much time to demonstrate that, but the models that we learn are very powerful, and they can be interrogated in many different ways to address different biological questions. As I briefly mentioned, we can use the models to identify novel interactions between regulators, or we can use them to identify the sequence preferences of uncharacterized TFs. We can also look at subsets of genes or conditions and identify the rules that are triggered by this particular subset of our data. We also had some very interesting findings regarding the differential usage of regulators or motifs in enhancer versus promoter elements. And if you want to find out more about the biological insights that you can get from such a model, I invite you to come by my poster and talk to me. So with that, I'd like to finish and thank my collaborators, Marco, who did an incredible job implementing this method, Seraphim, who is my advisor, Ansu, who is the inventor of the original Medusa method and was also very influential in this work. He also happens to be an assistant professor at Stanford, and he's hiring. And of course, I'd like to thank the entire Bajoglu and Kundaya labs, my sources of funding, and all of you for your attention.
So you looked at differential expression by taking all pairs of cell lines, or did you specifically look at pairs that are the most closely related so that you can sort of have more biologically meaningful differences?
No, we just considered all pairs.
Have you looked into sort of grouping them into some kind of tree of, differentiation based on either what's known or in an unsupervised way?
So I think this particular method can benefit from having different, different conditions, because in some sense, the more diverse your data are, the more there is to learn, right? If you have two very similar cell lines, you're not going to learn much by comparing them.
But I'm suggesting using the exact same data, using all of the data, and simply structuring the relationships in a more directed way, so that you're looking at specific pairwise changes rather than all n by n.
So the model is built based on all pairwise comparisons, but if you want to focus on a specific comparison, you can, after you build the model, you can sort of look at the particular sort of subtree that explains, this pair of conditions that you're interested in.
From the learning model itself, do you get a prediction of which ones are the most informative pairs?
The most
basically, do you find it's the most
informative
distant
pairs of
ones?
cell lines, you mean?
Yeah. Do you find that it's the most distant ones or the most close related ones?
I guess indirectly you could look at that by looking at which pairs of conditions tend to follow similar paths of the tree. Yeah.
Okay. Thanks again for a nice talk.
Okay.
Yeah, one more quick question, and just a comment that I'm happy to see Medusa live on. I wanted to get a sense of how much sequence space you're using for representing the genes. So you did say ChromeHMM enhancers and promoters, but how distal is the sequence?
So in terms of length, there's no fixed length, right? Because we just take the predictions, the predicted enhancers or the predicted promoters. So they tend to be, in the order of KB, but they vary. And
but how far away can the enhancer be to
so
the
for the
transcription
enhancers, obviously, there's always the problem of how you associate enhancers with genes. We consider enhancers that overlap genes, so it's kind of obvious which gene they regulate, and sort of proximal enhancers that are within 50 kb from the gene, and ignore the rest, basically.
Did you try restricting to DNase hypersensitive sites instead of ChromeHMM predicted enhancers?
No, we haven't tried that.
Okay. Thanks so much, and let's thank all the speakers from this session.